I would like to talk to you for a few minutes about what our capabilities are in the genomics core at UMass Boston. Next, please. I do want to point out that the majority of this equipment was purchased through funds from Mass Life Sciences, so thank you very much. Here I'm just diagramming the three basic areas that we cover in the genomics core: sequencing, nucleic acid integrity and quantitation, and uni and multiplex DNA and RNA analysis. Next. In terms of sequencing, we have sequenced everything from microbes through yeast, through rice, through lettuce, through lobster, through people. So we have sequenced just about every kind of organism there is out there. So it doesn't matter what kind of system or model system you're using. I'm sure we have some experience with it, or at least can figure out how to deal with it. We do high and low throughput sequencing, whole transcriptome, whole exome, whole genome sequencing, non-coding RNA-seq, ChIP-seq, long read-seq, single-cell-seq, and low input-seq. We are very good at troubleshooting difficult jobs. We work mainly outside the normal workflow of larger genomic cores. So if you have kind of a unique job, something that would require some troubleshooting, that's where we have expertise. And I think that's really kind of where we shine, and that's what our mission is largely focused on. You can see here we have an Illumina NextSeq 2000. That's like the latest model from Illumina. It's much cheaper to run this than some of the larger instruments, and I'll show you it has very good coverage. We have a 10x Genomics Chromium model, so we can do single cell RNA sequencing, an Illumina MiSeq for smaller jobs, and an Oxford Nanopore for long reads. Next. You can see here we do library prep services of all kinds of beginning nucleic acid substances, DNA, RNA, and we can actually purify down to very small types of sequences, like for miRNAs or small non-coding RNAs, if that's what you're interested in. In terms of the platforms we use, from the MiSeq all the way up to the NextSeq, we can sequence read counts that are from 1 million all the way up to 1 billion. So please, we can cover the gamut here, and also all kinds of insert sizes, as you might expect. As you can see here, I'm just showing the instrumentation again, just to give you a sense of the breadth, I guess, of the kind of genomics and sequencing services we offer. Next. If you do have large jobs, it's not that we don't do them, we do them. And we actually have a robot that can help us handle the library preps. This one has actually been designed to do that. Next. In terms of nucleic acid integrity and quantitation, we have your typical kind of instrumentation, the nanodrop, the qubit, and the Agilent Bioanalyzer. Next. The Agilent, we have both the Bioanalyzer, the lab on a chip, and the tape station. And this will allow us to look at your DNA, RNA, or protein and let you know how much you have and what the integrity is, so that we hope that you have decent quality, I guess, of especially nucleic acids. But honestly, we have been able to work with some fairly degraded and not so great in terms of optimization type nucleic acids fairly successfully. Next. We also have a couple different ways to do more targeted transcript analysis. Next. We have a Quant Studio 12K Flex instrument for polymerase chain reaction and quantitative RT-PCR. It uses high throughput 384 well plates, and it actually uses assay by demand probe sets, which allows you to not have to invest a lot of time into trying to figure out your own primer sequences, to be able to look at levels of transcript expression or levels of DNA quantity as an amplification or deletion. Next. We also have a Nanostring Encounter, which is a different kind of technology. It uses barcodes and kind of like homologous sequences at each end of your RNA or DNA to help you quantitate that and identify it. And actually what's nice is this is very good at multiplexing, so you can read up to 800 different transcripts in one sample and quantitate all of them with references therein, so you have something to count back to. Next. So by all means, check us out. There's our URL. If you go to umb.edu and type in genomics core, you'll get us. And there's our email right there. That's our core manager, Susan Padalano. She's actually a UMass Boston grad, so we're very proud of her moving way up to a manager of genomics core. We take vouchers and we use iLabs. So actually, if you go to our website and create an iLab account, it's very simple. You can see all the services we offer, how much it will cost. And you can contact us and we'll work with you on your project.